The process in which the bleeding is checked from a damaged blood vessel or injured blood vessel is known as hemostasis. It's a phenomenon to check the excessive bleeding. It's a precise and dynamic mechanism. It operates by the local as well as the neural myogenic central mechanisms by virtue of which the blood flow is regulated. And once bleeding occurs, it has to be stopped. Otherwise, a person may die due to bleeding. And this mechanism is said to be quite precise. Why? If it will occur more, then blood clotting will occur inside the body, which is also not required. So therefore, it is essential to understand this mechanism of hemostasis. Now the question arises, number 1 point, how this mechanism is initiated. Number 2, what are the responses of the damaged blood vessel? Number 3, when a blood vessel is damaged, what events occur in sequence? Number 4, how the blood flow is checked. Number 5, how that clot is stabilized. Number 6, how regrowth of blood vessel occurs which is already injured. Before answering these questions, it is also interesting to know why then a clot is not formed inside the blood vessel. So go back to my previous lecture where I have already explained why the blood flows in a streamlined manner inside the blood vessel and usually do not show the formation of clot. So today in this lecture, we are going to vividly discuss about the phenomenon of hemostasis, its mechanism and its steps. So before going into the details, please like and subscribe my channel. Let's start with the word hemostasis. Hemostasis is a phenomenon of, it is a check of excess loss of blood, or I can use another term, the process in which bleeding is stopped. It involves the local as well as spatial mechanisms. These local and spatial mechanisms operate in a specified manner, and that we are going to discuss. Here, 3 to 4 events occur in sequence. The number 1 is the local vasoconstriction. Vaso means blood vessels and it is a constriction means it is narrowing. After that, then there is a word known as platelet plug or clog formation. This is the second phenomenon. Then third, formation of a blood clot. That means a stable clot is formed at that point. And the fourth point is that growth of tissue along clot. That means the blood vessel which is injured, now it will show healing so that the blood vessel is completely healed from inside as well as outside. So these are the 4 phenomena: local vasoconstriction, which is followed by the platelet clog or plug formation, followed by the formation of the blood clot, and then formation of or the growth of the tissue. Let's draw a beautiful diagram to explain this mechanism. This hemostasis mechanism is mostly mediated by a definitive cell of the blood that is known as platelets. These are bigger cells while they are in origin, being known as the megakaryocytes. But during their origin, these megakaryocytes are converted into fragmented cells and they become smaller. And these cells are known as the platelets, the circulating platelets, which are fragmented. They are not nucleated. The cell organelles are also not defined that much. But these are the cells which are involved in the process of blood clotting. So let's see what I said, that this is a normal blood vessel. I am drawing 4 diagrams so that you can well understand. This is the continuous endothelial lining showing the endothelial cells which are negatively charged, so that the cellular surface and the cells which are flowing in the blood vessel are repaired. What then happens? Now, once there is an injury occurs here, once injuries occur, then the endothelium is exposed. Here, the endothelium is damaged, and the underlying collagen is exposed. Otherwise, we cannot understand the mechanism. And once this happens, here, this is that surface where the damage has occurred, and the blood is coming out here. So what happens during this time? The first is this A. To this, a response is seen, that is B. That is vasospasm or vasoconstriction locally, because this will cause this hole or this injured surface to reduce its diameter. Now the point is, this vasospasm is the first or the earliest event to occur, so that the excessive flow of the blood is little bit minimized and further events of the clotting cascade will start. This vasospasm occurs due to several mechanisms. Here I can write one by one. So this is the second mechanism, but the earliest to say after the collagen exposure. So what is that mechanism? That is, number one is the serotonin, which is released by the platelets as well as the damaged endothelium. There is a protein which is released by this endothelium, that is known as endothelin. It also plays a major role in the process of vasoconstriction. Then the third one is the local factors. The local factors means local myogenic factors. This is smooth muscles which are regulating that. Then also neural pain pathways. As injuries there, that there is a certain kind of pain pathways going on through the neurons. They are also the regulator of the vasospasm. Then both neurogenic mechanisms, they are also causing the phenomenon of this vasospasm. Also thrombostenin, this is a protein which is released from the platelet, that is also causing the vasospasm. So can you see how dynamic these phenomena are? And now you are seeing that there is a B mechanism is there, which is vasospasm. Next, coming to the dynamic mechanism, that is the third mechanism. Now the platelets which are flowing, these are the platelets which are flowing. Usually they are repelled by the intact endothelium. They are flowing. But once the endothelium is damaged, now this platelet surface changes. And that change causes the attraction of the platelets to the site of the injury and to attach. It's a cascade process. Once one platelet is attached, it will release something that we'll discuss, that will attract the other in sequence, and consequently a group of platelets, a mass of platelets will accumulate there. So what happens? Now here one platelet, second platelet, third, fourth, fifth, like this, number of platelets are clicking to each other. And once they click to each other, that word is known as the C. This is the platelet plug formation or platelet clog formation. You also use the word primary blood clot. Now, to understand this mechanism, what actually happened, that why this is there. So, intact endothelium, this point is important. Intact endothelium, it releases the prostacyclin, and this prostacyclin repels the glycoprotein of the platelets. Therefore, the platelets do not adhere. This is in normal blood vessel, do not adhere. And therefore, in normal blood vessel, it is a promising mechanism to keep the platelets away as well as free, so that a clot should not be formed. But when the blood vessel is damaged, we need to have a platelet plug so that the next processes of the clotting cascade will be initiated. So therefore, this point is important. But once the epithelium is damaged or endothelium is damaged, this causes, this point to be noted, this causes 3 to 4 modifications locally, those which will attract the platelets. Number one, the von Willebrand's factor. This von Willebrand's factor is released from endothelium as well as platelets, and it's present in the plasma. This is the factor which initiates the process of neutralization of this glycoprotein covering of the platelets. And what happens now, due to this von Willebrand factor, this is the von Willebrand factor, like this, they are being present at the site of injury. Just try to understand this point. And they make the point of contact of these platelets, and these platelets, they are attached. So this is the beginning of this platelet plug formation. Let us understand further in details. The detailing is that what actually happens. Now, as I told, there is the endothelium is already damaged. So first point is the von Willebrand factor. So see the diagram. This is that surface where the collagen is exposed. Now, this is the von Willebrand factor which is flowing in the plasma. This is that protein. Therefore, in the deficiency of the von Willebrand factor, the bleeding delays and sometimes blood does not clot because there is no formation of the primary blood clot. Now the platelet will come and attach. So this is, you can say, adherence of platelet, and this serves as a limiting step, this von Willebrand's factor. Then what will happen? Now 3 steps occur. Swelling of the platelets. Now the platelets will become swell. Number 2, there is membrane instability. The platelet membrane gets more fluidic and it becomes rough. The surface becomes rough. Rough platelet membrane or the surface. This is another point. Then the 4th point is that it forms pseudopore-like process. Pseudopore or false foot-like process. That means finger-like projections will come out. So now this platelet will become little bit swollen, irregular, and false foot-like processes start. As a consequence to this, now this will attach here. It will attach here. These are the platelets. They further will attract the attachment of other platelets to the site like this. Now they are all attaching. So these 4 fundamental points also result in the release of the compound adenosine diphosphate and thromboxane. You have to remember these are the points, thromboxane A2. And once ADP and thromboxane A2 is there, the platelet gets more activated. The platelets get activated, and this causes degranulation of the platelets. Degranulation. That means the granules which are inside the platelet, they are released. This is a calcium dependent mechanism. And by virtue of this, now the platelets are adhering to each other. And that's known as the platelet clog or the platelet plug or the primary blood clot. So what is the result of this? Now the result is gradually the platelets will come to the site and they will attach to each other. Now the third process will become, because now we have to deposit the fibrin here. Here the fibrin will be deposited like this. Then the fibrin will be polymerized and a stable clot is formed. So the third step, so first I said that there is a vascular spasm is there. Second, I told there is a platelet clog is there. Now coming to the third mechanism. Let's understand. So finally we can draw a master flow chart. So what is that point? That point is the C. That is fibrin deposition. And this fibrin deposition will occur when the fibrinogen is converted to the fibrin, and that process is known as the clotting cascade or clotting mechanism. That is my coming lecture. So in this is the clogged platelets which are attached to each other. Now these fibrin, they will accumulate here. These are the fibrin monomers which are stabilized in the presence of factor number 13, the clotting factor 13, that is fibrin stabilizing factor, and a stable clot is formed. This is stable clot. So this is the third point. Now 4 or 5 questions here. How a stable clot is formed? How fibrin is formed? What converts the fibrinogen, and what are these cascades? Why it takes some time? And once we will understand this, then we can understand what holistically has happened to this place where the bleeding is being checked. Then coming to the fourth and the last part. Now, this is stabilization of clot. What we are saying, clot retraction and growth of tissue, because it is already damaged. The stable clot is formed, no doubt. It will take another 30 to 40 minutes of time, and then the clot will be retracted, dehydration of the clot, serum will come out, size of the clot will be reduced, fibroblast will migrate to that area, the platelet will secrete a factor known as PGF, platelet growth factor, and endothelium also secretes a factor, endothelial growth factor, fibroblast growth factor, which will all together will cause a new tissue to be formed because of this fibroblast proliferation. And ultimately what will happen, now the damaged blood vessel will be healed like this. This is healed, now the blood is flowing. So this is a beautiful mechanism. As it is an introductory class, I told you the introduction, I told you the steps. In the coming lectures, we will go into the details. Every process need to be understand what actually happened and how it is happened. Now, before concluding the class, I will draw a master flow chart, and then we will stop this class, and in the next lecture will come for the mechanisms of blood clotting. I will put several question marks and which will be the answers in our next lecture. So now next see how sequentially these events occur. The first event is damage to the endothelium. That means the blood vessel is damaged. This causes the exposure of the collagen for local vasospasm or vasoconstriction. How this is? This is due to the serotonin which is released by the platelets, local myogenic mechanisms, or the neural pain mechanisms. Altogether, there is a vasoconstriction is there. This is local. Then what happens? The second one is platelet migration. Then second point, platelet activation. The third point, the platelet aggregation. The fourth point, ADP and thromboxane 2, A2, they will cause further activation of platelets. This will increase the adherence, and this is known as the platelet plug or the platelet clot. So this is the second part. When the platelets are activated, they release that master compound, the serotonin, which will further cause the vasoconstriction. The third point, fibrinogen. This will be converted to fibrin. This conversion is mediated by thrombin. How? Thrombin is formed. What are the mechanisms? What do you mean by intrinsic and what do you mean by extrinsic mechanism? That we'll understand. This fibrin get polymerized, and this polymerized fibrin is known as a clot. That means this primary blood clot is now converted to a secondary clot or stabilized clot. And then this clot is migrated by the fibroblasts, and there is tissuegenesis. That means the growth of the tissues or formation of the tissues or the healing. So all together these processes occur in a sequence. So with this particular lecture came to the conclusion. My next lecture is how the platelets get activated, what are the constituents they released. Second, how this fibrinogen is converted to fibrin and how this fibrinogen-fibrin conversion is mediated by thrombin, from where the thrombin come, what is the mechanism associated with the formation of thrombin, that we'll discuss. Then after this, we will try to understand how this fibrin is stabilized in the presence of factor number 13, how the clot is dissoluted, how the clot is retracted, how the clot is become stable, and then the complete flow of the blood is checked. Second, we should not forget that what is the role of von Willebrand's factor and why it is deficiency causes bleeding, because already I told that this is the plasma protein, this is the endothelial protein, this is the platelet protein which causes the platelet to adhere. So if this protein is being deficient, then the platelets will not adhere, and if primary blood clot is not formed, then secondary will not form. So this is the take home message. And the second point is that we should understand platelets are the regulator. So therefore, if the platelet number is reducing, normally 2 to 3 million per ml, if its number is reducing, then platelets will delay the process of clotting or the clotting will not occur. Thrombocytopenia is the term given to reduction in the platelet number, and the thrombocytopenia purpurea, a clotting disorder in which the stable blood clot is not formed and small size clots are formed in a disseminated manner. So we should understand this phenomenon in a sequence manner. So stay tuned for my next lecture where I am coming with these mechanisms which are not touched today and we will vividly discuss. So keep learning and stay tuned for my next lecture. Thank you so much.